And with that, I'd like to introduce our speaker for this evening. Her name is, I don't have to refer to my notes because it's right up here. She's an assistant professor, Rachel Gallery, in the School of Natural Resources and the Environment, and she's going to talk to us tonight about microbes, the little things that rule the world, microscopic life in soils. So please give a big round of applause, Dr. Rachel Gallery.
How's that? Good? Okay. Hi, everyone. Thank you very much for coming out tonight on this beautiful evening in December. Like Shepard mentioned, we're gonna talk about the little things that rule the world. So if you would, with me, if you would all inhale and then exhale. Thank you. Yes, yes, yes. And then touch your finger too. No, I'm just kidding. No. And bear with me, but the point that I'm trying to make here is that what we were just doing was we were inhaling oxygen and we were exhaling carbon dioxide. And we were using our lungs to do this. And we're doing it because it's a critical property of staying alive. And so my take-home message for this talk tonight is that we can think of soils as the lungs of Earth, of our biosphere. And the microorganisms, the billions of microscopic organisms that live in soil are inhaling oxygen and exhaling carbon dioxide to maintain their life. And through this process, they are sustaining us. And so really microbes were Earth's first ecosystem engineers. Right? They were the first colonizers of our planet, and all of biodiversity on this planet stems from microscopic organisms. And we're going to talk about some of that diversity and complexity tonight. So you may know, or may not, if you were at my colleague Craig Rasmussen's talk a few weeks ago, that this is the year 2015. This is the International Year of Soils. And that's really kind of an astounding and spectacular thing. And so things like for example, in the climate summit that's currently going on in Paris, most of the initiatives, either directly or even indirectly, they require the consideration of soils, and specifically the microorganisms in soils and the activities that those microorganisms are performing that are cycling nutrients and storing carbon. And so efforts like these and the Global Soil Biodiversity Initiative and the Earth Microbiome Project, these types of initiatives are highlighting the ecosystem services that soils and soil microbes provide. And so what do I mean by that term, ecosystem services? So the source of this quote is unknown, but I think that the sentiment resonates quite a bit. Man, despite our artistic pretensions, our sophistications, and many accomplishments, owes our existence to a 6-inch layer of topsoil and the fact that it rains. Right? So that puts it in context for us, doesn't it? Yeah, it does indeed. And so that is to say that ecosystems sustain societies, and they drive, excuse me, and create our economies, and it doesn't work the other way around. Right? So Franklin Delano Roosevelt knew this. He knew this in 1937 when he said, "The nation that destroys its soils destroys itself." But what is it about soil and soil biodiversity that's so important? There are many factors here on this slide that was just published this past week by Diana Wall and her colleagues in the journal Nature. That's talking about soil biodiversity and human health specifically. So outlining for us some of the services that soil biodiversity provides. And so if we start sort of in the middle of this web, and we're going to define all of these terms in a moment, but we can think about species richness and what that means in terms of how an ecosystem works. And then what that means in terms of what we care about, things like our disease load and the diseases of the plants that we eat. The air that we breathe and the water filtration services that soils provide, all of these things can be tied back to what microorganisms are doing in soils. And when we think about this system and how it has been operating for millennia and how it will continue to operate, there are important feedbacks. Some of them are things that we have control over directly, such as how we manage our land, And some of those are things that we have control over perhaps more indirectly, such as our contribution to climate change. And so, as I've already alluded to, greater than 99% of Earth's biodiversity is microscopic, right? It's invisible. That's 5 million trillion, 5 times 10 to the 30. I don't really know what that number means, right? But it's kind of an impressive number, so we put it out there. So this is 5 million trillion bacteria. And that doesn't even include all of the microscopic archaea, the viruses, the nematodes, and the fungi that make up what we're talking about when we talk about microbial diversity. Here in 2015, most of us have heard of and are aware of microbes. At least we've heard about them, maybe in the context of disease, well, certainly in the context of disease, but then perhaps and hopefully also in the context of the benefits that they can provide. And it's true that the Earth, as well as ourselves, we are teeming with microorganisms. Some of you may have heard of the microbes that live in and on our bodies. And together, the mass of that microbial consortia that lives in and on us weighs about 3 pounds, so essentially about the same weight as our brain. It says the microbial communities on us, that is their equivalent weight. So we are teeming with these microbes, and they influence all aspects of our physiology. They help us digest our food. They prevent us from certain infections. They adjust our immune systems, and the same can be said for the crops that we eat and rely on. And the same then can also be said for the soil microbiome. So just to kind of throw out another hopefully astounding example, in this 1 gram of soil, if you were to pick up a gram of soil from pretty much anywhere, even in a desert or a tropical rainforest, or go out to the Sonoran here, pick up that gram of soil, and you will find perhaps up to 1 million different types of microbes in that gram. And then more astoundingly, if you were to actually count the number of cells that were in that gram of soil, you'd come up with a number that is greater than the human population here on Earth, right? And that's just in that gram of soil. And they're doing a lot of work. And let's talk about that. So first, let's kind of get at some definitions, because I think that's important. What do organisms in soil look like? When we talk about soil biodiversity, let's first be all-encompassing, and let's include everybody. So we have here some really nice photos of some fungal hyphae that are growing from a pine root. We have some nematodes. We have the tardigrade, who's been in the news a lot recently. The genome was sequenced. We have mites and different earthworms and ants and columbella and moles, right? So we have some macroorganisms in here as well. And those macroorganisms are incredibly important in terms of how they influence the physical structure of soils, and so how they influence soil aeration and basically turning over in soils, moving oxygen and water through that system. But for the most part, the real drivers in soil function are happening at the microscopic level. So the diversity and our path towards understanding these microbes and viewing them as more than just, agents of disease started about 400 years ago. But it's really only been gaining momentum in the past few decades as we have advanced our tools for figuring out how to observe them, how to quantify them, how to identify them and compare them, and understand what each and every individual microbe is doing. And these tools have been improving at an exponential rate. So speaking of tools, how were these microbes discovered to begin with? Like I said, this happened about 400 years ago. Antony van Leeuwenhoek. We call him the father of microbiology. But what he was so he was Dutch, and he worked with drapes. He was a draper. And we can attribute the discovery of microbes to someone who was working towards improving the version of the microscope at the time in order to better distinguish thread quality, right? So this is a microscope at the time for scale, so that would be about 3 to 4 inches, and you would hold it up to your eye like this. And he was working on improving a series of lenses so that he could really adjust that focal length and get at really significant levels of detail. And he was doing this, like I said, so that he could better understand the thread quality in the cloths that he was working with. And so, to use the metaphor that when you have a hammer, everything looks like a nail, he basically went around and was like, well, what else can I see? Right? So he would take a little bit of pond water, look at raindrops in his microscope. He would sample the saliva from his mouth, and if you think about sort of oral hygiene at the time, you can imagine sort of a wondrous array of diversity that might have been there. And what he did was he saw these things that were in there, and they were moving around, and they were certainly alive, so he thought. And he called them animalcules. And you have to remember that he wasn't a scientist, and so he started drafting these really long and intricate letters to the Royal Society of London, and basically was met with nothing. No one believed him. Why would they believe him? They had microscopes. They hadn't. Their microscopes were of much poorer quality, and they hadn't been able to see what he could see. So, over a series of years, he was finally vindicated. And here are some of the drawings that he was able to discern from the microscope images that he was seeing. And so we now attribute him as the father of microbiology, right? All because of some thread quality distinguishing. And so this is how microbes were discovered. What are they? Where are they? This is the evolutionary tree of life that was first proposed by Norm Pace. And so we have different lineages, different branches on our tree of life. They include the bacteria, the archaea, and the eukarya. And let's talk about this in a little bit more detail. All of the microscopic organisms are highlighted under these bubbles. So all of the bacteria and the archaea, these are these single-celled organisms. They're all microscopic. And even if we move into the multicellular branch of the tree of life, which includes us out here, the majority of these branches are microscopic multicellular organisms. And there's only a few branches that include us and our sort of more recent relatives that are not, right? So the majority of the tree of life itself is microscopic. Which actually brings us to a really important question, which is, what is a microbe? And the actual definition of a microbe is not very, I think, interesting or useful. It simply means a microscopic organism, something that can't be seen with the naked eye. So that doesn't give you a lot of information about what it does, right? And so it's kind of a meaningless term unless we put it into context. So for example, the term microbe could refer to cyanobacteria, so these photosynthetic organisms that would be functionally equivalent to land plants in terms of the process of photosynthesis. Microbes could also refer to heterotrophic fungi, so fungi that are involved in decomposition. For example, that I'm showing you here is an aspergillus. So the interesting thing about aspergillus is that there are so many different life history strategies of all of the different species that are within this group. And so they have a lot of different roles in an ecosystem, much like we do. Of course, many microbes cause disease. And so this malarial plasmodium, it's microscopic, it's vectored by a macroscopic mosquito. And then finally, there are top predator microbes, if you will. So these are microscopic nematodes. These are voracious consumers of other nematodes. They're very ingenious and, like I said, voracious, much like a killer whale, right? And so we have a lot of functional similarities between these microbes and these macrobes. And yet, it is still useful to think of them as a community or a consortia. They have specific traits, but at the same time are working often together within this consortia to cycle nutrients or to drive some particular ecosystem process, like storing carbon. And what I'm going to do for the rest of the talk is run through two examples for work that's going on with my research group in the School of Natural Resources and the Environment. I have an excellent group of women, and I'm going to specifically be showing some work done by our graduate students, Noelle Espinoza, Martha Gebhardt, Maggie Murphy, and Dawson Fairbanks. And so I'm going to use this figure to sort of introduce two different but related concepts. And the first would be that of microbial biogeography, or the distribution of microbes around the globe and in different biomes. And the second will be to talk about how we can identify and measure the activities of these microbes in soils. So what they're doing in terms of cycling phosphorus or nitrogen or storing and releasing carbon. Okay, so we do not need multivariate statistics to know whether we're walking in a tropical rainforest or in a boreal Arctic forest, or when we're walking through a grassland, or when we're walking through a desert scrub. Right? We can be walking through these ecosystems, and we know where we are, largely based on the dominant vegetation, right, the life history characteristics of those plants, the structure of those plants' communities. It can be pretty obvious. So yes, there's going to be a little bit of overlap in terms of species and traits, but nevertheless there are distinct communities when we're talking about tropical rainforests versus taiga or boreal forests versus grasslands and shrub steppes. Can the same be said for the soil bacteria in these biomes? The answer is, for the most part, yes. And so I'm going to walk you through this figure and just point out a few of the important highlights. So here what we've done is each one of these points on this figure represents a soil collection. So soil was extracted from the earth, and then from that soil we extracted the DNA, all of the microbial DNA, and we're able to identify the different bacterial taxa within that soil. And so each one of these points represents one of those soils. We can look across these two axes, and then we note for ourselves that for the points that are closer together, those represent communities that are more similar to one another. And the points that are further apart are far more different. And so when we look at it this way, we can see that there's a lot of overlap in terms of the bacteria in tropical forests versus in boreal forests. And on the first hand, you might think, well, that doesn't make so much sense to me intuitively. Those are very different biomes with different precipitation regimes, different mean annual temperatures, different vegetation. But if you think about it from the perspective of a microbe, it might make a little bit more sense. These are forests, and so they contain and are dominated by woody vegetation. And so they contain a lot of the resources and the food, the substrates that the microbes use and that they eat. And so from a microbial perspective, even though they are tropical and boreal forests, they are both forests. So, thank you for the question. These axes are non-dimensional. And so the analysis that we ran is called a non-dimensional multiscaling analysis. And so you don't necessarily look at the values on these axes and derive information from them. You use them to compare within this space, the assembly. And what would happen is it would be required to do a number of other analyses to try to figure out what was driving the patterns that you see here. And the students, this paper that I am showing you these results from, they did do this. And one of the things that they found was, for example, one of the main explanations for what's driving the pattern that we see across this x-axis is the change in pH from the different soils. So the pH in these sagebrush steppes here in Utah are far more basic, more alkaline than the pH of the soils in these forests. And then if we were to think about some of the variables that describe this axis, those would include things like soil water and to a large extent a number of other trace elements, but mostly soil water. And so when we were looking at this, we can orient ourselves and say, all right, this axis is mainly telling us that pH is pulling these samples together or apart, and this could be driven by soil water. So thanks, that was a great question. So we can see this overlap in these forest communities and this distinction over here in Utah. So that's given us sort of a snapshot that we're going to return to in a minute, but a snapshot about the distribution of these different bacteria in these soils. But what are they doing? And how are they cycling carbon and nutrients? And how can they help in terms of ecosystem recovery, for example, in the face of a disturbance like a wildfire? And so that's what we're going to talk about next. And so wildfires are, of course, incredibly important here in the Southwest, but also globally. And here we have fire return intervals that are increasing in frequency and intensity with some devastating consequences. So let's go to the Jemez River Basin in the Valles Caldera in New Mexico. This is one of the areas where the Critical Zone Observatory has a research site. So this is just a map to kind of orient us. The elevation in this area, sort of on the eastern part of this Redondo Peak, is about 3,000 meters. And the data that I'm going to be showing you are taken from this sort of area here, this basin. So and those points represent sampling locations. This is an image I pulled from Google Earth of the site. So there's that kind of basin outlined for us here. This was taken 1 year before the Thompson Ridge fire, so in May of 2012. And you can see it's pretty lush, pretty green, but there's also a lot of variability on the landscape. So we have areas where we have denser tree cover and areas that are far more grassy. And then in 2013, Thompson Ridge fire blew through and burned about 10,000 hectares of forest. And a few weeks after that fire, the students went out and sampled once it was safe. But before we get there, this is just another image that I took from Google Earth to show us 1 year after the fire what this landscape now looks like. And so obviously this is a big difference. Let me actually just toggle through and show you. So quite green, quite dead. And so this would be all standing dead tree or now even fallen dead. And yet we still also see some green on the landscape, right? So we see some areas, specifically within that kind of convergent zone of the landscape where we might have a lot more surface water availability. We still see in these areas at least that it's a little bit greener. Maybe this is due to quicker plant recovery. It also might be due to the fact that there was a less severity in terms of the burn on the landscape at that position. And so just as we can look at a map and we can look at the distribution of vegetation on that landscape, what we were trying to do was use a similar sort of analogous approach for looking at microbial activity on that landscape. And one of the ways that we can do this is we can look at the different food sources that microbes are using. And I'm going to show you a map here. This is talking about microbial carbon activity. And then I'm going to show you another one that's dealing with phosphorus. And in this case, this map is showing us the activity of breaking down essentially like a sugar molecule. And so the take-home points from this map are that the warmer colors, so the colors in red and yellow and orange, these are high activity areas. And then the cooler colors, so the blues and the light greens, that's low activity areas. And so when we look on this map, we can basically say, all right, there's something happening on this sort of partitioning of the landscape where we have much higher carbon activity, whereas over here, that activity is much lower. Right, so that's one pattern that we can pull out and then start to look at all of the other data that exists through the Critical Zone Observatory to try to draw correlations and then hopefully identify mechanisms to explain the patterns that we're seeing, both in terms of the activity of microbes and of plants. So this is our example for carbon. And this is taken, I should say, in the first 0 to 50 centimeters of soil. So we're talking about surface activity here. And if we then compare that, so that's that carbon map that we were just looking at, we compare that to a phosphorus activity map, one of the things that should hopefully strike you is that the patterns are different, right? And so whereas we have, in this case with phosphorus, much higher activity in that zone on the landscape where we saw much greener vegetation at the time, right after that burn. If we just kind of tie this now to going out and taking actual photos of the areas that were sampled, there are a couple of things that we can identify. For example, this particular area here was very severely burned. And so we see low activity, both in the case of microbial turnover of carbon, but also mineralization of phosphorus. Low activity in both of these places. And we can probably attribute that to the severity of burn in the landscape. Whereas when we look over here at this area, which is very bright red for both phosphorus and carbon activity, we also now see that the vegetation has really come back. We can also see standing water. So we see what's happening in the soils is that we have much more waterlogging, which means that you have conditions where you have very low oxygen availability, so these anaerobic conditions. And what's important here to note is that those physical processes and chemical processes in the soil can be selecting for particular groups of microbes over one and another. So fire is stressful. Certain microbes would have been killed, others will be stressed, and yet others might even thrive in these different environments. And so what we're interested in doing is taking what we see in terms of the activity in the system and then trying to relate that to who's actually there and what they're doing. And so this is the final sort of data slide that I'll show. And what I'd like to point out is that we see these shifts in the diversity and also the similarity of communities as we move from one site to the next. So here on the x-axis we have 4 different sites. So those would be, these 4 different grams of soils that we've taken all of that bacterial information from. On the y-axis is the relative abundance in terms of percentage, in terms of all of the different bacterial taxa that might be within the sample. And so there are a couple of patterns that just strike us immediately. First is that down here with these dark blues, those are unclassified bacteria. And that shouldn't be surprising to us because there is so much that we don't know about bacterial life. And so this basically is showing us a huge diversity of unknowns within our sample. And there's a lot of value in these unknowns in terms of bioprospecting and looking for potential new medicines and new advances that we might work with in terms of drought stress and drought tolerance for our crops. So there's a lot of information that might be gleaned from understanding these sections of the bars more. And the other important pattern to draw from this is that in this particular site, the number of unclassifieds is much lower than in site number 4. And then we see a much different pattern happening if we go and we move up and look at the proteobacteria, so these red here. Now the proteobacteria are a group of bacteria that many of them are involved in nitrogen cycling, right? And so then we might be able to draw some inferences about the rate and the nitrogen cycling potential of site 2, where we have a large number of proteobacteria, relative to site 4, where we have a much smaller number. And we can go on and so forth. So for example, the next group on our figure are the acidobacteria, which are kind of a recently discovered clade. And the reason for this is because they are incredibly prevalent within soils, but they can't be cultured. And before we started using these tools where we would take some DNA out of the soil and sequence it and identify it, the only way to identify these microbes was to try to grow them out on different types of media and identify them that way. And so these acidobacteria were largely unknown because we couldn't grow them. And we can see that they constitute a huge proportion of the community. And then finally we have actinobacteria, which they are usually dominant in soils. They're really common in the soils. And just as important are all of these very, very rare taxa that we see up at the top of our figure. Many of them we don't really know what they do, but we know that their populations wax and wane, And if we take snapshots like this at one period of time, and then go back and take it over a period of an entire year, incorporating all of the different seasons at a particular site, we can see this really beautiful sort of turnover in the organisms and their relative abundances over time. So why do we care about that, and why is that interesting? Well, it brings us back to the role of these soil microbes in ecosystems and the services that they provide. And these services, they include the ability of ecosystems to potentially recover from disturbances like fire, the ability of ecosystems to prevent disease outbreaks, both in plants, but then also in humans, and the ability to grow food and other natural resources for ourselves. And so this potential lies specifically within the soil biodiversity that we see, so this microbial life in soils. So with that, I'd just like to thank you very much for your time, and I'd be happy to take any questions.